Assalamualaikum and welcome. I bid to Dr. Hanis and my fellow friends. We are from Group 10 presenting on cross-presentation in non-antigen presenting cells. Before any action can take place, an intruding tumor cells must be present in order to stimulate a signal. The first signal requires the recognition of the tumor antigen. However, tumor cells are not professional antigen-presenting cells, so they cannot migrate to the lymphoid organs to activate the naive T cells. Instead, specialized dendritic cells at the tumor site ingest tumor antigens through endocytosis. These dendritic cells then migrate to the secondary lymphoid organs carrying the tumor antigens with them. Inside the dendritic cells, the antigens are processed via the endogenous pathway and presented on MHC class I molecules. This unique process is known as cross-presentation. It allows the dendritic cells to activate naive CD8 positive T cells, which would otherwise be impossible due to the tumor cell's limited role in immune signaling.
For a naive CD8 T cell to become activated, it needs signal 2, the co-stimulatory signal. This happens when CD28 on the T cell binds to B7 molecules on the dendritic cell. But here's the challenge with tumors. They often don't trigger strong innate immune responses. So, dendritic cells that pick up tumor antigens aren't fully activated and express low levels of B7. This means the CD8 T cell may not receive enough stimulation and stays anergic or inactive. This is where CD4 helper T cells step in. Once activated, they express CD40 ligand, which binds to CD40 on the dendritic cell. This boosts the dendritic cell's ability to activate CD8 T cells by increasing B7 levels and cytokine production.
After signal 2, the T cell receives the green light to become activated, but to fully differentiate into a powerful killer T cell, the CD8 T cell needs a third signal, and that's where the cytokines come in. The dendritic cell secretes interleukin-12, a cytokine that promotes the expression of CTL effector molecules such as perforin and granzymes. In addition, CD4 helper T cells, along with the CD8 T cells, secrete interleukin-2, a key growth factor that supports CD8 T cell proliferation and survival. Other cytokines such as type 1 interferons and interferon gamma further assist in the overall differentiation process. Thanks to the coordinated cytokine support, the CD8 T cell differentiates into a functional cytotoxic T lymphocyte, or CTL, equipped to detect and destroy abnormal cells. After differentiation, the CTLs leave the lymph node and travel through the bloodstream guided by chemokines to the site of infection, or in this case, tumor. There, they recognize target cells displaying the specific tumor antigen on MHC class I molecules. Once engaged, the cytotoxic T cells will carry out their effector mechanism, which will now be explained in detail.
The first mechanism in granule-mediated killing is the recognition of the antigen. The cytotoxic T lymphocyte, or CTLs, will begin by scanning cells for danger, and these tumor cells will display a peptide antigen on its MSC class I molecule. The CTL recognizes it using a T cell receptor, or TCR. Then the CTL will begin to approach the tumor cells to form binding of antigen. The TCR binds to the peptide MSC1 complex, while CD8 binds to the site of MSC class I molecule. It is to strengthen the interaction between the cells. Then LFA1 binds to the ICAM1, forming a strong adhesion. For the second mechanism, which is immune synapse formation, a close contact zone called the immune synapse now forms. This seal-off zone allows the CTL to safely deliver its killing agents only to the tumor cells. This minimizes harm to healthy neighboring cells. So now, moving to the third mechanism, which is the polarization of the CTL. Inside the CTL, the microtubule organizing center, or MTOC, rotate toward the immune synapse, and special killing granules are then transported to the synapse, following the microtubule tracks like a guided missile system.
The cytotoxic granules then fuse with the cytotoxic T cell membrane to release its contents, perforin and granzymes, into the immune synapse through exocytosis. Then, perforin facilitates the delivery of granzymes into the cytosol of the tumor cell. Granzymes activate caspases that initiate the process of apoptosis And this can occur within 5 minutes. After apoptosis, cytotoxic T cells will detach from the tumor cells. The perforin granzyme-mediated killing serves three main advantages. Cytotoxic T cells are highly antigen-specific and only eliminate the target cell that presents with tumor antigen on their MHC1 molecule. The immune synapse formed creates a close contact region that prevents harming nearby cells. Apoptosis does not trigger inflammation like necrosis.
Cross-presentation of antigen is important as it activates CD8 plus T cell against viruses that don't infect epices. Same as explained before, some viruses infect body cells such as liver cell. These infected cells can't activate CD8 plus T cells because they don't express co-stimulatory molecules, and since they are not epices, they can't go to lymph nodes. Cross-presentation allows dendritic cell ingest infected cell and do something special, which they load the viral antigen on MHC class 1, even though they didn't get infected. And now, the DC can fully activate CD8 plus T cells, which go destroy infected cells. Same goes for tumors. Disease can cross-present tumor antigen to launch anti-cancer CD8 plus responses. Without cross-presentation, viruses and tumors could escape. This is make sure they don't. That's all from our group. Thank you for listening to our presentation.